Happy day to all and welcome back to the M.O.P classroom. I am Dr. Sara Jane Monica, Assistant Professor, Department of Food Science, and I am here to talk about the different ways by which bacteria can be classified. So, microorganisms are defined as minute, unicellular or multicellular organisms that are invisible to the naked eye and hence can be viewed only under the microscope. The different microorganisms that are existing in the universe can broadly be categorized into two different classes. The first one is called as prokaryotes and the second one is called as eukaryotes. In case of prokaryotes, structurally, you will not find a nucleus along with other membrane bound organelles in their structure, whereas in case of eukaryotes, there is going to be a presence of a well defined nucleus along with the presence of other membrane bound organelles such as mitochondria, lysosomes, Golgi apparatus and endoplasmic reticulum. Bacteria are examples of prokaryotes, whereas viruses, fungi, algae and protozoa come under eukaryotes. Moving on to the classification of bacteria. Bacteria can be classified based on 6 different aspects such as based on nutrition, oxygen requirements, shape, arrangement, temperature and staining technique. The first one is classification based on nutrition. So, based on nutrition, bacteria can be broadly categorized into two main classes. The first one is called as autotrophs and the second one is called as heterotrophs. Autotrophs, they are able to synthesize their own nutrients required for cell division, and depending upon what source they rely on to derive their nutrients, autotrophs can further be categorized into 3 different classes. The first one is called as phototrophs. So, this particular group of bacteria, they are able to utilize sunlight and get the energy. Those that derive energy from chemical reactions are called as chemotrophs, and those which has the ability to reduce organic compounds and derive nutrients required for cell multiplication are referred to as lithotrophs. The second one is called as heterotrophs. Heterotrophs cannot synthesize the nutrients and hence they have to depend upon other sources to get the nutrients required for growth and multiplication. Now, depending upon what source they rely on to derive the nutrients, heterotrophs can further be categorized into 3 different categories. The first one is called as saprophytic bacteria. This group of bacteria derive their energy from dead and decaying organic matter such as fruits, leaf, vegetables and fecal matter. The second one is called as parasitic bacteria. They get the energy from the host in which they grow, and one specific character of this particular group of bacteria is they can either be harmful or harmless in nature. Harmful in the sense they have the ability to cause some kind of disease, and disease causing microorganisms are referred to as pathogens. Example is Bacillus anthracis, which is the one responsible for causing a disease called anthrax in livestock. And the third one is called as symbiotic bacteria. They live in association with other organisms and in turn they are said to be beneficial to the host. Example is nitrogen fixing bacteria. Now, this particular group of bacterium, what it does is it has the ability to fix free atmospheric nitrogen into nitrogenous compounds which are utilized by the plants, and the plants in turn they provide the nutrients required for their growth and multiplication, which means that both of them get benefited in one way or the other. The second one is classification based on oxygen requirements. Now, as human beings, all of us require oxygen for our survival in this universe, but whereas in case of bacteria, certain group of bacteria requires oxygen for its growth and they are referred to as obligate aerobes, whereas another set of bacteria called as obligate anaerobes, they cannot survive in the presence of oxygen. The third one is called as facultative bacteria. Now, this particular group of bacteria, it can adapt itself to the present situation, meaning it can either live in the presence of oxygen or in the absence of oxygen. The third one is classification based on shape. So, based on shape, bacteria can be classified into 5 different groups. Oval or spherical shaped bacteria are called as cocci, rod shaped bacteria as bacilli, comma shaped bacteria as vibrio, rigid spiral shaped bacteria as spirilla, and the last one is called as spirocetus. They are said to be spiral shaped, but at the same time they are coiled in nature. The fourth one is classification based on arrangement or how they appear in nature. The first one is called as diplococci. Di means to say, so they appear or occur as pairs. Example is Streptococcus pneumonia, which is responsible for causing pneumonia and meningitis in human beings. Then there is something called as tetrads. Tetrads means they appear in groups of 4. Sarcina appears in groups of 8. Streptococcus occur in chain form. Example is Streptococcus pyogenes. And the last one is Staphylococcus, wherein they appear grape-like clusters. Example is Staphylococcus aureus. This bacterial strain is one of the most common strain responsible for causing food poisoning in human beings. The fifth one is classification based on temperature. So, depending upon what kind of temperature is required for bacterial growth and multiplication, we have 3 different categories. The first one is called as thermophils. So, this particular group of bacteria has the ability to grow at extremely high temperature ranging between 45 degrees Celsius and 120 degrees Celsius. Then the second one is called as mesophiles, which prefer or grow at optimum temperature ranging between 20 and 45 degrees Celsius, meaning in the presence of high temperature or low temperature, they cannot multiply. The third one is called as psychrophils. Psychrophils can grow only at the presence of very low temperature below 20 degrees Celsius. The last one is classification based on staining technique. So, the staining technique is called as gram staining technique, and gram staining technique categorizes bacteria into 2 groups, either gram negative or gram positive bacteria based on the permeability of cell wall and cell membrane. Now, gram staining technique is said to be one of the most important steps involved in identification and characterization of bacteria. This technique involves 4 simple steps. So, this figure tells you the 4 steps involved in the staining technique. So, once you get the bacterial smear, first thing we have to do is add few drops of primary staining solution, that is crystal violet, followed by addition of few drops of gram cyanide solution. After that, you will end up adding few drops of decolorizing agent, which could either be acetone or ethanol, followed by addition of the secondary staining solution, that is safranin or neutral red dye. Now, once this experiment is over and when it is viewed under the microscope, if it is gram positive bacteria, they appear purple in color, and in case of gram negative bacteria, they appear pink or red in color. Now, let us examine the reason as in why gram positive bacteria appear purple in color, whereas gram negative bacteria appear pink or red in color. Now, structurally, if you see, the cell wall of the bacteria is made up of a mucopeptide called as peptidoglycan. Peptidoglycan in turn is made up of 2 subunits called N-acetylmuramic acid and N-acetyl glucosamine, which are linked together by short chains of peptide. Now, when you look at the composition of gram positive bacterial cell wall, 50 to 90% of it is made up of peptidoglycan, and the purple line given in the figure indicates the peptidoglycan. So, gram positive bacteria, it has a thick cell wall, and that is the reason why it is able to retain the primary stain, and hence it appears purple in color. On the other hand, gram negative bacteria, it has a thin cell wall and only 10% of it is made up of peptidoglycan. That is the reason why it is not able to retain the primary stain solution color. Instead, it is able to retain the counter stain solution. Examples of gram positive bacteria includes Staphylococcus aureus and Streptococcus pneumonia, whereas Salmonella typhi and Shigella flexneri comes under gram negative bacteria. Salmonella typhi, we all know that this is the bacterial strain responsible for causing typhoid fever in human beings, whereas Shigella flexneri causes an infection called as shigellosis, which affects the intestine. Now, in terms of practical aspects, if you want to do studies focusing on characterization and identification of bacterial strains isolated from any particular food sample, then you will be in a position to do couple of tests such as simple staining technique, which will give you information about the shape and arrangement of the bacteria. Using gram staining technique, it will tell you whether it is a gram positive or a gram negative bacteria. Using a growth tolerance test, you will be able to find out the optimum temperature at which that particular bacterial strain grows, And lastly, you will do a culture test to find out what is the mode of nutrients. So, to do all this test, definitely you have to have little bit knowledge on different ways by which bacteria is classified, and that was exactly what that was taught in today's lecture session. Thank you.